Hi, my name is Estie Rose. I'm a genetic counselor at Montefiore Medical Center and for the Program for Jewish Genetic Health of Yeshiva University. One of the main goals of our program is to educate people about how being Jewish can put you at risk for certain genetic diseases. There are many genetic conditions that have been identified in individuals of Jewish ancestry, but before going into detail about this relationship and about the diseases in general, I think it's important that we start from scratch so that you can have a clear understanding of a few basic concepts in genetics. With a firm background, it will be easier for you to grasp more complex ideas in upcoming lectures. In this lecture, we'll be covering some basic vocabulary terms and also inheritance patterns. So let's start with vocabulary. Some of these concepts might already sound familiar to you. The first term is the word cell. Our bodies are made up of millions of cells. Cells are the basic building blocks of all living organisms. For example, many liver cells together will make up a liver. Many heart cells together will make up a heart. And many skin cells will make up skin. All of these organs put together make up an organism, like a human being. The cell contains different structures, or organelles, and each have a unique function. The structure we're going to focus on today is the nucleus, because that is where our genetic material is stored. So inside the nucleus of each and every one of our cells is a set of chromosomes. Chromosomes are made up of tightly packed DNA. This is the picture of a chromosome set in one cell. What you see here is 46 chromosomes, or 23 pairs. For each set, one of the chromosomes came from the mother, and the other chromosome came from the father. The first 22 pairs are just numbered 1 through 22. The final pair determines the gender. 2 X's is a female, and an X and a Y is a male. The picture you see here is a normal female. Any number that is not 46 is not normal. So here's an example of a chromosome set in one cell where there is an extra chromosome 21. An extra chromosome 21 causes Down syndrome. Having this extra 21 in each and every one of this individual cells leads to a whole range of physical and mental changes. Down syndrome is an example of a chromosomal disease, and it cannot be repaired. Now that you know what a chromosome is, the next term you should be familiar with is gene. Genes are stretches of DNA that make up the chromosome. So imagine that a chromosome is a string of pearls, and each individual gene is a pearl. We have about 10 to 20,000 genes in each and every one of our cells. Each gene codes for a different protein, and each protein has a different and special job in our bodies. So what makes each gene unique? What differentiates one gene from another is the unique sequence of bases. There are 4 bases, and each gene has a different order of these bases. If even one of these bases is wrong for whatever reason, it can alter the functionality of the gene. If the function of the gene is altered, then that change in bases is called a mutation. If the gene still works, then that gene is called a benign variant. It's totally normal to have benign variants, and it's actually what makes us all different and unique from one another. Another term that has become popular in the genetics world is genome. The genome is all of the hereditary information of an organism, so it includes both the coding and the non-coding parts. The coding parts of the DNA are the genes. The non-coding parts may be regulatory. We don't know the function of many of these non-coding regions just yet. The Human Genome Project was completed in 2003 after 13 years of very hard work. It was a huge undertaking where the scientists were able to determine the sequence or order of all of the bases in all of the genes of the human organism. They also mapped out all of the human genes, so we know exactly where each and every gene is and what it's supposed to look like. The next big wave in genetics and determining why certain genetic diseases happen is to look at a patient's full genome. This is called full or whole genome sequencing. Here are 2 more words to add to your genetics dictionary. Your genotype is your full genetic information. It's how your genes look. This shouldn't really be changing over time unless your genes become mutated by some external things such as radiation exposure. Your phenotype is your observable characteristics. It's how you look. Your genotype is the major influencing factor in the development of your phenotype, but it's not necessarily the only one. How you look will also depend on environmental factors and how those factors interact with your genes. So here's a picture of two eye colors. One genotype will translate into brown eyes, while another genotype would be associated with blue eyes. Here's where things start to get a little bit tricky. Just because somebody's genes look a certain way, it doesn't guarantee that they'll exhibit the phenotype that's associated with it. Penetrance refers to whether someone who carries a particular genotype will also express that phenotype. If every person who carries a certain genotype exhibits symptoms of the disease, then that disease is said to have complete penetrance. An example of this is achondroplasia, which is an inherited form of dwarfism. The inheritance of a specific mutation in the FGFR3 gene will always result in the disease achondroplasia. However, some diseases have reduced or incomplete penetrance. For example, if a disease is said to have 95% penetrance, that means that out of 100 people who have the same mutation, 95 of them will develop the disease, but 5 of them will not. Many cancers with a hereditary basis exhibit incomplete penetrance. A great example of this would be retinoblastoma, which is a rare cancer of the retina that primarily affects children who carry a mutation in the RB1 gene. Interestingly, not all people who carry this mutation suffer from retinoblastoma. Two sisters could inherit the same mutation from their parents, and one might be affected by the disease while the other is not. While measures of penetrance focus on whether or not a disease is expressed at all, the degree to which it's expressed can also be measured. For instance, if one patient is suffering from a disease, they might have very severe symptoms, whereas another patient who has the same genotype might have very few symptoms. Meanwhile, a third patient might not have any symptoms at all. This phenomenon is described as variable expressivity. A good example of expressivity is the genetic disease neurofibromatosis. Neurofibromatosis is a disease caused by a mutation in the NF1 gene. These mutations cause certain cells in an affected individual's nervous system to grow into tumors called neurofibromas, which can appear as cafe au lait colored spots or bumps under the skin. These tumors can result in skeletal abnormalities and severe neurological problems. Not all people who carry the mutated NF1 gene will actually exhibit symptoms of neurofibromatosis. Some people just have discolored skin, while others could have very severe neurofibromas. Now that we know what a gene is and how its expression can be variable, let's move on to discussing how genes are implicated in diseases. Some diseases manifest because of a mutation in one gene, and others are due to a combination of genes and environmental factors. It's much easier for geneticists to test for monogenic diseases because we know which gene to analyze for mutations. There are several thousand monogenic diseases that have already been described, with more to follow. Multifactorial diseases, on the other hand, can result from the combined effect of alterations in multiple genes. Alternatively, they can develop when there is a genetic predisposition and also lifestyle or environmental issues that spur it on. A great example of a multifactorial disease is diabetes. People tend to have diabetes because they have a combination of certain mutated genes and also lifestyle factors that could spur it on. The last concept that you should know about is the founder effect. The founder effect is the enrichment for certain variation that occurs when a new population is established by a very small number of individuals from a larger population. For example, the Jews of Eastern Europe, or Ashkenazi Jews, have lived in small insular communities for hundreds of years. Mutations in genes were present in these communities, but they weren't very common. The communities changed over time. There was migration outside, there was isolation, and decreases in size. As a result, the mutations that had been rare earlier now became more prevalent. This is why the frequency of Tay-Sachs mutations is particularly high in the Ashkenazi Jewish population. And there are plenty of other diseases that are also common in this population because of the founder effect. But Ashkenazi Jews are not the only population to have high carrier frequencies for this reason. In fact, the French Canadians, the Irish, and the Louisiana Cajuns also have high carrier frequencies for Tay-Sachs because of the founder effect. In addition, sickle cell disease is more common in the African American and Hispanic populations. Thalassemia is common in Southeast Asians and Mediterraneans. and cystic fibrosis is common in Northern European populations. The next part of our lecture will focus on how genetic diseases are inherited. Each genetic disease has to start somewhere, so let's go through all of the possible ways that the disease could be passed on to a child. Each and every genetic disease is classified as either recessive, dominant, X-linked, mitochondrial, or de novo, or some combination of these. Let's start with autosomal recessive. Autosomal recessive diseases happen when both parents are carriers of a genetic mutation and both of them pass it on to a child. Carriers of autosomal recessive diseases have one working copy of the gene and one non-working or mutated copy. Carriers do not show any symptoms of the disease, nor will they ever get the disease, because having one working copy is enough to protect them from the disease. But when two carriers of the same disease have children, each one of them has a 50-50 chance of passing the mutation on to their child. When this happens, the child is actually affected with the disease because they have two mutated copies of the gene. There's a 1 in 4 or 25% chance with each pregnancy that a carrier couple would have an affected child. Examples of recessive diseases include Tay-Sachs, sickle cell disease, and PKU. Unlike autosomal recessive diseases, autosomal dominant diseases develop because one working copy of the gene is not enough. So if somebody has a mutation in a gene implicated in an autosomal dominant disease, then they'll actually be affected with the disease. Someone who has a dominant disease has a 50-50 chance of passing on that mutation to their child. So each of their children have a 50-50 chance of actually being affected with the disease. Examples of dominant diseases include Huntington's disease and Marfan syndrome. X-linked diseases occur when the mutated gene is on the X chromosome. Remember what I said before. Females have 2 X chromosomes, and males have 1 X and 1 Y. If a female has a mutation on one of her X's, she still has another X to compensate, so she won't be affected with the disease. She'll be considered a carrier. However, if a male has a mutation on his only X chromosome, he would actually be affected with the disease. That's why X-linked diseases more commonly affect males. If a woman is a carrier of an X-linked disease, she has a 50-50 chance of passing on the mutation. If she passes it to a girl, then the girl will have 1 X with the mutation from her mother, and her other X from her father will not have the mutation. She'll be a carrier. If the carrier woman passes the mutation on to a son, then his only X will have the mutation and he'll be affected. Examples of X-linked diseases include Fragile X syndrome, which is the most common inherited form of mental retardation, colorblindness, and some forms of albinism. Less common are genetic diseases that are maternally inherited. Like I said before, most of our DNA is found in the nucleus of the cell. However, what I didn't tell you is that there's also a small ring of DNA in the mitochondria. When a baby is being formed, the mother's egg is what contributes the mitochondria, not the father's sperm. So if there's a mutation in the mother's mitochondrial DNA, then that mutation will get passed on to the child. A father cannot pass on a mitochondrial disease. An example of a mitochondrial disease is Leber's hereditary optic neuropathy, which causes vision loss. Mitochondrial diseases are not as common as recessive and dominant diseases. De novo means new. So these conditions occur because of a new mutation that neither parents have nor transmitted. This mutation will happen either in the parent's egg or sperm and not in the other cells of their body, which is why they don't actually have the diseases, or the mutation can happen once the egg is already fertilized. When a child is born with a de novo condition, most of the time it's unlikely for the family to have another affected child. Thank you for listening to Part 1, Basic Concepts in Genetics and Inheritance. Stay tuned for Part 2, Genetics Professionals and Legal, Ethical, and Social Issues in Genetics. For more information about our program or for questions, please feel free to contact me.